Yes.
Alright, so in the previous class, I think we left off at ABO blood grouping, right?
Yeah.
Did you have any doubts up until now?
No.
Alright, so today we're going to start with the dihybrid cross. So basically, when just like how monohybrid cross was there, so we took one character into consideration, right? And studied the inheritance of that one character. But when two characters are taken into consideration and their inheritance is studied, then we call it as a dihybrid cross. So basically, two characters, how are we taking it? For example, you're taking two things such as seed shape and seed color together. So that's two characters, right? Earlier, we took only plant height. At that time, let's just say if we took plant height as well as flower color, that would have been dicross. So a cross was made between a pure, meaning a homozygous round yellow seeded plant, which is a dominant character, alongside a wrinkled green seeded plant. So round yellow will be represented as capital R for round, capital Y for yellow, because that's a dominant. Small r for wrinkled, and small y for green seeded plants. Now from them, ultimately the gametes would come out to be R, capital Y, and here small r, small y. Now when you cross them together, the F1 generation would come out to be the hybrid, which is capital R, capital Y, small y. Now, in order to find out the F2 generation, you have to do selfing, isn't it? So before you do the selfing, you have to take out the gametes. What are the possible gametes that would come out of the F1 generation? So how do we take it out? We take it out from this particular cross. So capital R with capital Y, that's gamete number 1. Then capital R with small y, that's gamete number 2. Then small r with capital Y, gamete number 3, and small r with small y, gamete number 4. So these are the possible gametes that are there. Now there will be two sets obviously for reproduction. One will be, yes?
9:3:3:1.
That's the ratio.
Yeah.
So then you take the two sets. Like for example, this is one set, cross it with another set because you're basically multiplying the male and the female together, like crossing the male and the female together, fusion and all of that happens. So now this board that you see, this checkered that you see is a Punnett square, with the help of which we find out the possible genotypes. Now, how do we operate it? You take the capital R, cross it with the capital R, capital Y.
Yeah.
Then again, you take this one, cross it with small r, capital Y. Then again with capital R, small y. And lastly, small r, small y. So you crossed it with all of them. And then same way for small r, capital Y, you cross it with all of them. So you have to take one, cross it with all four. And ultimately, you will be left with all of the possible genotypes that are there. Now, can you tell me the phenotype of this first one?
It is round and yellow.
Second?
It is round and yellow.
Third?
It is round and yellow.
Fourth?
Again round and yellow.
Fifth?
It is round and yellow.
Sixth?
It is wrinkled and yellow.
Seventh?
It is round and yellow.
Eighth?
Wrinkled and yellow.
Ninth?
It is round and yellow. Round and yellow. It is round and it is green. And then it is round and it is green. And then it is round and it is yellow. It is round and is and wrinkled and it is yellow. And then it is round and it is green. And then it is now the one is wrinkled and it is green.
So out of
Last
all of the possible phenotypes, only one of them is a pure, you can say, recessive condition. Rest all of them are heterozygotes. And ultimately, when you count the ratio, that the phenotypic ratio comes out to be 9:3 is to 3:1. Clear?
Yeah. Yeah.
Now, there is another law, the third law, that is the law of independent assortment. So, according to this law, if two pairs of contrasting characters, meaning 4 traits in total, are present, each pair will segregate independently from each other, meaning the round segregates from yellow. It's like there will be no blending in between them. The round will be shown like separately, yellow will be shown separately. So all of the expression of all of the genes will be happening like that. That is law of independent assortment. Then comes multiple allelism. Now, multiple allelism is a phenomena which tells us that for one gene, usually 2 alleles are present, right? Dominant and recessive, isn't it?
Yeah. Yeah.
But when there are more than 2 genes or 2 alleles for one gene, then we call it as multiple allelism. Okay?
Yeah.
Now, there was this question that out of all of them, out of all of those options that you see, which of the phenomena are present in ABO blood group?
Oh, I know this answer. Wait, ma'am, is it multiple allelism? Think.
See, the options are also there. So you see the option.
Oh, like, like, okay,
You have to
more.
choose multiple from them.
Okay. Mm.
All right, let me simplify it for you.
Um, ma'am, won't it like show only in one, maybe like codominance? Just codominance?
Yes,
Isn't it just codominance?
codominance is there, 100%. Apart from
Yeah.
that, there is also multiple allelism.
Multiple allelism. Yeah, those two are there. I told multiple allelism and codominance.
And
But
normal
then,
dominance is also there.
huh? Acha. Huh?
Because
Okay.
if this is the case, what does it show? Dominance.
Okay, 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 okay. Okay.
Clear?
Yeah. Okay.
Then comes the next topic, that is chromosomal theory of inheritance. Is this chapter completed in your school?
Not yet.
Not yet. I got your message, the one
Yeah.
that you sent. So in that you were asking me what?
Ma'am, can you like teach in the class, and then at the end I'll take the screenshots and write the notes later or something like that. Or last 10 minutes I can write the notes because we have only 1 hour, so that's why.
Okay, no problem. All right, so now talking about the chromosomal theory of inheritance. Now see, can you tell me the significance of chromosomes?
It carries genes.
It carries the DNA,
And
which carries the
DNA
gene. Hmm.
genes.
Basically, the chromosomes, they are the physical carriers of the DNA, right? The DNA, they are physically present on the chromosome. Now,
Mm.
according to Mendel, he started working on the pea plant, but now, yes, this theory that Mendel started to work on the pea plant in the year 1865, but his work was unrecognized until
90.
1900s. Why?
Yeah.
Because first of all, there was lack of communication, because at that
Mm.
time the media was not so advanced. Apart from that, he used mathematics and statistical knowledge in the field of biology. And
I remember one. Like he used to like that time many things are not yet discovered, so he used to call the things as factors, and that's why he couldn't really like tell the people what was what in his things.
yeah, he did not have any proof because nothing was discovered at that point of time. So
Yeah.
he called them as factors.
Mm.
Lastly, chromosomes and all of these things, they were not discovered at that point of time.
Mm.
So in the year 1900, there were three scientists, Hugo de Vries, Correns, and von Schermat. So they independently rediscovered Mendel's results on the inheritance of character. Like all three of them worked separately, and they came on to the same conclusion that Mendel came on to. And at
How
like
did
by
they find it? Like, what was their experiment? What did they
they
do?
also were studying inheritance, so maybe they took some animal or maybe they took some plants and like that, and they all came to one conclusion because apparently the inheritance happens the same way no matter what the organism is. So even
No,
though they
because I didn't understand that honeybee one. Some honeybee one was there.
honey what?
Uh, yeah, like
Oh,
the
haplodiploidy and all of that.
I didn't
We'll
understand.
come to it. Like, so
Okay.
your teacher is not teaching you serially.
No, no, no. She taught this way before, but I didn't understand that time, like completely. So,
Okay. So, by 1902, the chromosomes were also discovered. So, Walter Sutton and Theodore Boveri, they were the two scientists that actually studied the pattern or the behavior of the chromosome. And they
mm,
said that however the chromosomes behave, that is how the behavior of genes work.
mm,
And they used the chromosome movement to explain the law that Mendel had already given. So
mm,
according to them, the pairing and separation of a pair of chromosomes would lead to the segregation of a pair of genes they carry. So if the chromosomes got segregated or separated, so each of the chromatids, whatever genes they were carrying, they will also get separated.
mm,
They'll not stay in touch, they'll not stay connected. Now, the knowledge of chromosomal segregation that happens during the cell division, along with the principles that Mendel had already laid the work for, this combined thing gave the chromosomal theory of inheritance.
mm,
Now, what are the features? According to the chromosomal theory of inheritance, the chromosomes, they retain their number, structure, and individuality. For example, if we talk about the genes, the DNA, when the DNA is passed on from one generation to another generation, it retains its structure, it retains its integrity. It doesn't change, not even one tiny bit. Same goes for chromosome. The chromosome number, the chromosome structure, their integrity, and their individuality do not change one bit. Right? And
mm,
it is like you can say hundreds and hundreds of generations, but it will not change. They like the two, whether we are talking about the genes or the chromosome, they neither get lost nor do they get ever mixed up. Yes, they
mm,
stay the way they are. This is the first point.
mm,
Now, both the chromosomes as well as the genes, they occur in pairs. in the somatic cells. So in the case of the body cells, they are present in a pair, meaning two sets. That is what we call
Like
as
they come
diploid.
together.
Hmm?
Like one comes from each parent like that.
No, we are talking about somatic cells, the body cells, not the gametes.
Okay.
But the body cells, they are diploid. Why are they diploid? Because they have two sets present. Whether it is the two sets of genes or the chromosome, it will be like both of them appear in pair. Now the
Okay.
two
So,
alleles of a
so there are 4 in total.
yeah.
Okay.
The two alleles of a gene pair are located on the homologous sites on the homologous chromosome. See, what happens? Homologous means at the same site in one chromosome. Like for example, if this is one chromosome, it is a homologous chromosome. These two for each other are non-homologous chromosomes. So when we talk about the two alleles of a gene pair, meaning the two possible traits, they are located on the homologous sites on the homologous chromosomes. Clear? Homologous sites on the homologous chromosome. Now both
Same.
both the chromosome and the genes, they segregate at the time of gamete formation, such that only one of each pair is transmitted to the gamete. Because we know that
Can you explain the first one?
homologous site on the homologous chromosome. See, homologous site means at the particular site on that particular chromosome, the two of the alleles will be present. It will not be the situation that one will be at this tip, the other will be at below.
Oh, okay. So like both of them will be like this. The alleles will be this like over here in the top part,
Yes,
right?
yes, yes.
Okay.
Meaning, basically, whether we talk about the gene or the chromosome that the genes are present in, it will be at one particular site and that's it, not several places. One
Okay.
site for one gene. Clear?
Okay. Okay.
And during the gamete formation, obviously, half of the chromosome goes to either of the gametes, right? So each pair is like only one of the pair is transmitted to the gamete through this meiosis, that is reduction and division. So this was the chromosomal theory, right? Theory. It's like nothing has been proved. Huh?
Can you explain the each pair is transmitted to a gamete?
See, for example, when gametes are made, okay, so gametes are made like initially the cells are diploid, right?
Yeah.
And always, like I've always said, that for the formation of gametes, they'll always be haploid.
Yeah.
If this cell is forming two gametes, so both of them will be haploid. So it's not
Yeah.
that the gamete is getting both the sets that the parent cell has. It's only getting either of the pair. It can be any, but one of the pair. This is what they're saying.
Okay.
Both chromosomes and the genes, they segregate. Segregate means separate at the time of gamete formation, such that only one of each pair is transmitted to the gamete. Always remember it as a
Can you write 2n and then put it in haploid and then just for understanding? Yeah.
Now this was the theory. Okay. Basically, of the top of their head, they have said this. But the proof for this has not been given at that point of time. Now, the experimental verification of this theory was given by Thomas Hunt Morgan, T.H. Morgan. He worked on Drosophila melanogaster, which is called as a fruit fly. Okay, now again comes that why not any other animal? Why only this one? Right? So the reasons,
Short
again,
cycle.
the biggest reason is that you can grow
Variety.
Drosophila on like synthetic medium, like you can grow them in lab. Most
Yeah,
organisms
and
you
they
can't
don't
utilize.
require, yeah, and they don't require that much of like attention and like,
Yeah, they don't require that much care.
yeah,
Their lifespan is very short. They just live for
very
2 weeks.
short.
That's it.
Mm.
And once they reproduce,
And
within those 2 weeks, they produce hundreds and hundreds of offspring. So that means
and
more and more.
they also have a lot of varieties. Like they don't just have one or two, like many of them. I think that's one of
Variety
the.
doesn't matter here. The more important thing is that once they reproduce, they produce a lot of offsprings. More
Mhm.
number of data, more number of observations. So that will give a better result. Also, in them, the male and the female flies, they are easily distinguishable. You can see the
Oh
male
yeah,
and
the
the
red eyes
female.
and the white eyes.
Hmm.
That I think.
What is this phenomena called as? Wherein you can distinguish male and female?
Mm. There is a phenomena. Hetero something.
It is called as sexual dimorphism.
Oh yeah. Okay. Okay. Okay.
You remember Animal Kingdom?
Yeah. Yeah.
So here the males, they are smaller in size. The females, they are larger in size. And that's how you can make out which one is which. Also, they have a very tiny number of chromosomes. They just have 4 pairs. So their this thing, their somatic cells
Six,
will be having 4 pairs.
three. Uh,
Somatic
three of
will
them.
have 4 pairs, whereas
Three of
out of them, half of
three of them. Three
huh?
of them are three of them are autosomal, and then one is sex chromosome.
Yes. Yes. Any doubts about that?
Nope.
Now, while T.H. Morgan was studying about the this inheritance, chromosomal theory of inheritance, like the experimental proof, so he came across 2 more things, that is linkage and recombination. Now, linkage is the physical association of the genes on the chromosome, like how tightly the genes are present on the chromosome. If the
Oh yeah,
genes are
yeah.
present
Okay.
very tightly, what will happen?
The recombination will be very, very less. Very,
Yes.
very less.
What is recombination?
Like uh new variations in present in the offspring.
Exchange of genetic material, particularly.
Okay.
Okay, meaning that uh, tightly located gene will not be inherited that easily. The loosely one will. So the
We'll have it.
generation of non-parental character, right? The crossing over, that is recombination. Basically, the exchange of genetic material. It happens during the crossing over stage. If you remember packaging of meiosis,
Yeah, yeah. Prophase one, right? Yeah.
now, the sex-linked genes, they were studied by the Morgan. Out of the 4 pairs that Drosophila had, out of them only the sex-linked ones were studied, not the autosomes, meaning not the body cells or body chromosomes. So out of the 4 pairs, 3 of them were autosomes, meaning they were the body chromosomes. And 1 pair was allosome, that is a sex chromosome. So Morgan, he carried out a dihybrid cross in the case of Drosophila, wherein he crossed the yellow-bodied and white-eyed female with brown-bodied and red-eyed male.
Okay. Wait, wait. Yellow bodied with the white-eyed female. Okay. And then
Body
brown
is
body.
yellow, eye is white. Brown is
Huh.
body, red is eye. Okay, so he crossed them together, and much to his surprise, he saw that the ratio at the end of the dihybrid cross, it did not come out to be 9:3 to 3:1.
Hmm.
It wasn't. Why? Basically, to his, you can say, understanding, he said that, okay, maybe some genes, whether it is the color of the body or the eye color, it may be tightly linked on the chromosome. And since it is tightly linked on the chromosome, they show very low recombination and they do not express that much in the offspring.
Hmm.
But if the genes are loosely linked, they show high recombination. So the recombination for the tightly one is 1.3%, whereas the recombination percentage for the loosely one, like loosely linked one, is 37.2. Clear?
Yes.
Any doubts?
No. Ma'am,
Hmm.
I wanted to tell you. one minute. There was a question related to this. Ma'am had asked, the frequency of a parental form is higher than 25% in a diploid, in a dihybrid cross. What does that indicate about the two genes?
Frequency of a
Frequency of a parental form
parental form.
is higher than 25% in a dihybrid cross.
Hmm.
What does that indicate about the two genes?
The frequency of, say it again, please.
The frequency of a parental form is higher than 25% in a dihybrid cross. What does that indicate about the
See,
two genes?
basically, if the frequency is higher than 25%, frequency means occurrence, right? How many times that trait is occurring in the offspring. So when we talk, you want to note it down?
Yeah, correct. Yeah. Ma'am, you know how I wrote the answer? How I made up the answer. You want to listen?
Please.
Okay. So I wrote this indicates that the dihybrid cross is obeying the law of independent assortment,
Hmm.
where its proportion is 9 is to 1. Hence, uh, if the alleles are close together, then the recombination for the next generation will be less in percentage. But if the alleles are far from one another, hence loosely located, they will show recombination. And then I drew the allele ka part. See,
That's correct.
like this. Like closely packed and then loosely linked.
Correct. This is what it signifies. Okay, if the frequency comes out to be higher than 25%, right, that means that there is the association of the genes on that particular chromosome. Right, and always as a fact, as a rule, the tightly linked ones would show lesser recombination, the loosely ones would show, you can say, more or better recombination. Clear?
Now, my question is, if they had given any other percentage, then we still have to write both the loosely and the tightly packed because we don't know what is coming, right? The Punnett square itself will have a lot of things.
Yeah, you have to write both because in the cross, there is no set possibility of one thing only. Because there is a possibility of both. It's all probable what will come in the offspring. So you have to write both, no matter whatever percentage it comes out to be.
Yeah. There's one more question. Can I ask? There's just one more. Two heterozygous parents are crossed. If the two loci are linked, what would be the distribution of phenotypic features in F1 generation for a dihybrid cross?
Two heterozygous parents are crossed.
Hmm.
Then
Are crossed. If the two loci are linked,
two loci are linked, okay.
what would be the distribution of phenotypic features in F1 generation for a dihybrid cross? Is this like the same answer or do I have to change
See,
it?
they are also talking about the locus itself, right?
Same, right? Like alleles, and then you just tell the location of the alleles and blah blah blah, and then you just finish it.
Yeah, you send this question, okay? You send this question to me. I'd have to see a better explanation because I'm not getting how to explain this to you. So you
Yeah,
send
she
this question.
got these from last year's boards.
It's good. It's a nice question. You send it to me.
Yeah. Yes, ma'am. I sent in the Learnivio.
Okay. Yeah, I'll see that question and I'll get to you with the answer. Okay.
Yes, ma'am.
Now, moving. Are there any more questions?
There is one more. It's just that name the scientist who proposed the chromosomal theory of inheritance.
Who did?
Boveri and
Saturn and Boveri.
yeah.
And who gave the experimental proof for the chromosomal theory of inheritance?
TH something.
T.H. Morgan. Thomas
Morgan.
Hunt Morgan. Okay.
Yeah.
Now, Alfred Sturtevant used the recombination frequency to measure the distance between the genes and mapped their positions on the chromosome. What he did, he saw that, okay, the recombination frequency, how much or how many times is that particular recombination percentage is happening again and again, and he used it to measure the distances between the genes, sort of mapping the genes on the chromosome. He wanted to find out which gene is present at which position on the chromosome. So when this mapping was done for prokaryotes, the unit that was used was map unit. And when the mapping was done for the eukaryotes, the unit was used was centimorgan.
I didn't know that. Ma'am did not teach that.
I think we have progressed a lot in the chapter. Almost coming to the near end. Almost, not completely. Where are you at?
She's at sickle cell anemia. Just finished sickle cell anemia.
Sickle cell anemia. Oh, then you're really far.
Yeah.
There's a lot of things then. So you
Yeah.
want to go along with her? Your exam is in May, right?
Yeah.
So
We should go
why
see her, right? I don't know.
yeah, that why are you in such a hurry? I don't understand. The more
Actually, okay, so the thing is, April
you want to
8th,
be
no.
like top in the class.
Yeah, that is there. That I'll do, inshallah. But
No, not that. Like while she's teaching, you want to know all of the things. That's what you want to do.
kind of.
Hmm. What were
Yeah.
you saying?
No, not only that, April 8th, from April 8th till April 13th, I will not be coming for classes. So it's a school trip. So they're gonna take us again for another place. So
Oh,
yeah.
wow, nice.
Yeah.
But still, I have covered a lot of things today. Do you think your brain can handle more? I'm asking you, I'm ready to teach. I don't have any problem. You are the one who has to learn.
Mm-hmm.
If you can, I'll do one more topic and then you write. Because you know,
Okay.
it's a lot for today.
Okay.
Because you grasp quickly. So in that way, I go through the topics very soon. Right? The time
Yeah.
that I would have taken to teach one topic, I have taught you 7 topics in that time. So
Okay, I didn't even realize. Okay.
So, all right. So now the next topic is this thing,
Sex
sex
determination.
determination. Now, sex determination is the process, the whole entire analysis through which how the sex of the offspring would be, right? And in different organism, it is a different thing. Now, see, you know that in humans, the female is XX and the male is XY, right? This much you already know. This phenomena of having the two of the same kinds of chromosomes or two of the same kinds of sex chromosome is called as this thing. This is
Homozygous.
home. This is called nothing. The phenomena of having two different kinds of sex chromosomes in one individual. We call it as heterogamety.
Yeah.
Heterogametes. Okay. Now, heterogamety means that once this individual will be contributing to the offspring, it will make an X gamete and the gamete carrying Y. Okay. Now, this heterogamety can be found in females and it can be found in males. Depends upon the organism. For example,
How in females they don't have heterogamity, right?
In humans, they don't, but in other organisms, they do.
Oh,
In
okay.
other organisms, there are females that decide the sex of the baby. But
Yeah,
in
the
human,
chickens. I mean, the hens.
but in
Yeah.
human, they don't. Now,
Yeah.
in turtles, the sex is determined by the
But
incubation
in
temperature.
temperature of water. Ma'am, I have a question regarding this. Yes, okay. So see, we know about turtles, right? Do crocodiles also have the same thing? But I checked in Google and I found out that crocodile eggs, I don't know, they told that incubation water something. So,
Hmm,
I mean,
I'll have to
even my
check, but I
teacher,
don't think so. But they have clidoic eggs, so I don't think so. Let me see.
because my teacher also told that no, but then I checked in Google, then she's like, okay, I'll confirm and I'll get back to you. And then, well, I never asked her again.
Hmm.
They
In
do, don't.
in crocodiles and alligators both, the sex
Ah,
determination is temperature dependent and not by chromosome.
yeah. Yes. Okay.
Meaning eggs that are incubated below 30 degrees Celsius, they will be females. Above 34, they'll be male.
Thanks. Yeah. Okay.
Okay, so turtles, crocodiles, and I'll write it here.
Alligator.
Turtles and crocodiles, basically the major reptiles.
Reptiles. Yeah.
Clear?
Yeah.
All right. Now, in the case of Drosophila, birds, humans, butterflies, grasshoppers, honeybees, the sex is determined by the gene. Now, Henking, in the year 1891, he discovered the X body, that is the X chromosome. Okay. Now, the production of two different types of gametes by one individual, we call it as heterogamety. Now, heterogamety can be there in the case of male also and female also. Stephen, these two names, I think they're a little extra, they're not in NCRT, but still you remember that Henking discovered the X body, Stevens
Yeah.
discovered the Y chromosome. And Wilson and Stevens said that X and Y are the sex chromosomes. Okay?
Yes, sir.
Okay, wait. I have one question that you should do. Related to the monohybrid cross. It's a very simple, cute kind of cutie question. We'll do that. Okay, there is the question. There it is. Can you see the screen?
Not the first one, right? Okay. Mm.
This is the question. A blue colored flower plant denoted by capital B, is crossbred with that of white flowered plants, small b. Now you have to answer these three questions. You can note down this in your notebook because it's a nice, it's a very tiny, like I said, it's a cutie question.
Ma'am, can I tell the answer? For the state the color of the flower, I think what would happen is there will be one which is blue, one which two which are light blue, and one which is white. Is it like that?
Did you make the cross?
No, I'm guessing because I
No,
think
make
this
the
is.
cross. Always make the cross.
Uh,
Because then your answer will change. Make the cross.
mm, mm, mm. State the color of flower you would expect in the F1 generation. Okay, in the F1 generation, all will be.
What were your answers?
Yeah,
Blue,
forget that.
light
Forget that.
blue.
Yeah, never mind. No, no, no. That is the F2. We'll see. We'll see. What is what percentage of white flower plant in F2 generation? Okay. Hmm. F2 generation, we need only a Punnett square because we can't do with the thing, right?
So you have to find out F2 generation first, then you can take out the percentage.
Yeah. Okay. I think it will be. No, I'm not guessing anymore. Maybe.
What is your guess?
No, 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 no. Forget that. I'm not doing that. Um, the F2 generation.
Hmm.
Um, um, so is it like, am I doing selfing or am I doing done?
What did you do in the first one? Tell me that.
That is capital B, small b. I crossed it. I got all blue.
So you got, what is the genotype of the offspring that you got? What are the
Uh,
genotypes?
capital B, small b.
Then all of them are capital B, small b, right?
Small b. Yeah.
So they are all heterozygously blue. Now for
Huh.
F2 generation, what do you have to do?
I have to do a Punnett square.
You have to do a
Right.
Punnett square, but what? How will you get to that point? So what?
Exactly. How do I get to that? So is it with like okay, I'll take like in the top part it'll be capital B small b, capital B small b. And then in the lower part, is it like one capital B, one small b?
Okay, this is a monohybrid cross. Don't complicate it. What do you have to do? You have your capital B, small b. You have these offsprings, right? Now, in order to find out the F2 generation, like I've said, what you do selfing. So you cross them together, what do you
Although
get?
I'm getting everything blue.
How are you getting everything blue? Find out
Although
what
I
are the
don't.
possible gametes from here. You have capital B, small b. The
Oh,
possible
wait. Stop. 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 Okay, okay, okay. Stop, stop, stop. Yeah, yeah, yeah. No. I'm stupid. Wait, 1 minute. Yeah, 1 minute. 1 minute. Okay, yeah. So I'm getting blue, and white.
Hmm.
Yeah.
So what is the
So
percentage of white flower?
the white flower is 1.
So percentage? 1%.
So I don't know.
No, dear. See, I'll tell you. See here. This is what you've got in the F2 generation.
Yeah.
Capital B, capital B, capital B, small b, capital B, small b, and small b, right? So
Yeah.
in order to find out the percentage in any of the situation, what you're going to do, since they are asking about the homozygous recessive, so how many of them are homozygous recessive? 1 upon the
Oh,
total number of gametes into 100. And that is how you're going to get 25%. Even if you
Mom,
look
I
at
have a
this,
question. So aren't they all supposed to be 25%?
yeah. But what if I ask you that what is the percentage of heterozygous dominant blue plants?
Heterozygous dominant blue plants. Okay, 2 by 4 and 2 by 4 into 100, correct?
What is the
Uh,
percent then? 50%.
um,
Hey,
huh,
no. What is?
huh. Okay. Hmm.
Lastly, the ratio 3:1 that you already.
Yeah.
Any doubts?
Yeah. I like this question. Okay. No.
So this was a very tiny miny question, and I wanted you to do it. This is actually from class 10.
Oh.
And I will make you practice dihybrid cross questions in the next class. Okay, so before the next class comes along, you have to come prepared with your concepts about phenotype, genotype, percentage, then crossing, Punnett square, selfing, all of those terminologies should be clear. Okay,
Selfing is just
selfing is
is
self-fertilization.
only self, right? Yeah. Selfing is okay.
Nice.
No,
What happened?
I did one mistake.
Hmm.
I did Punnett square instead of like selfing, in the F2 generation.
See, Punnett square will also suffice, but you have to do it in the right manner.
Right manner, exactly. Right. Yeah, I took like both. I took capital B, small b, capital B, small b, all of them.
That's why. You have to take
That's it.
out the gametes, okay?
Yeah.
Then, oh god, my screen is stuck. Did you understand the whole entire concept?
Yep.
Can you see the screen?
Yeah, I can see now.
What is the last thing that you've written?
Mm.
I'll come with a lot of questions in the next class. Okay, so you be prepared.
Okay.
We will solve many, many questions. Because this
Yeah.
chapter, as many questions as you do, it's better.
Yes.
Where are you at? I'll share the whole entire PDF of this chapter with you. Okay. After the class is done, I'll share, but you will not misuse it. Meaning you will write your notes. I'm saying after I share the notes of this chapter with you, like today only, I'll share the whole entire notes. But I'm saying that
Okay.
you will not misuse it, meaning you will note down, not use the PDF as your notes.
No, I'm too lazy to open the app and do that. Yeah.
Good, good. We like that kind of a lazy. Okay.
Yeah.
What were you writing in the previous class? Where were you?
Monohybrid cross.
That was completed.
No, explains the inheritance of one gene from there.
Okay, you write down. We just have 5 minutes left. You write down.
Mom, I'll take a screenshot or something.
I'll
I'll
give you the
write it.
whole entire notes. No? So what's the point? You write a little bit and then the class will go. because
Okay.
I'm not going to teach more than what I've taught today. Because that's
Yeah.
more than enough for one class. And in the next class, I don't want this chapter to end like that. You know,
Oh, okay. Yeah. Okay. Yes, sir.
also, Rida, the picture that you've sent, that's your handwriting. Wow, beautiful.
Oh, yeah. Sorry, my mic was off when I was answering like
That's
that.
your handwriting.
Yeah.
How come
It was
I've
a
never
mistake.
checked your paper and saw this handwriting?
You saw way long back.
Very nice.
The
Mashallah, very beautiful handwriting. Very,
the
what do you call it? The early
cursive.
times newspaper kind of handwriting. Very nice. The letter kind of.
Yeah. Ma'am, can you scroll down? Okay, so now.
Okay, let's just keep it till here only. I'll be sending
Yeah.
you the PDF, okay?
Yes,
And
ma'am.
you can read from my notes. And I'll bring and be prepared with dihybrid and monohybrid cross, because I'll